So hello students, welcome to Vedantu NEET English. This is Basavaraj Sir, your biology master teacher. Quickly let me know in the chat how is everyone doing. And if you're doing great, moderate, you're doing really amazing, quickly let me know in the chat. And also while you're at it, also let me know in the chat if I'm audible to all of you and the beautiful presentation behind me is visible to each and every one of you. Quickly let me know in the chat, all of you. Yes, good afternoon each and every one of you. Now students, this is a part 2 of NCERT line by line of the principles of inheritance, right? In the first part we did the basics, and students, I would really recommend to watch the first part. Why? Because in the first part we did something called as the basics, monohybrid cross, dihybrid cross, incomplete dominance and codominance. All the following topics were covered in the first part. So I would highly suggest watching the first part and also continuing with the second part. Why? Again, students, in the second part we'll be doing something called as chromosomal theory of inheritance. Then we'll be looking at something called as linkage and recombination. Then pleiotropy. Then polygenetic inheritance. After that we'll be looking at what is the meaning of mutation, what is point mutation, what is frame shift mutation, and basically how does a mutation play a vital role. After that we'll be doing something called as your pedigree analysis, and finally all the Mendelian and chromosomal diseases. Now that is today's platter. Okay, so all the topics will be covered in today's class. Students, quickly fire emoji in the chat if you're excited to learn this concept. Why? Because we will be solving a lot of PYQ. See, this chapter, the concepts are very easy to understand, but when it comes to questions, the questions are very crafty. When I say crafty, they are small, little tricky they are. So if you want to solve those tricky questions, watch the video till the very end. You will be able to solve all the questions which we are doing in today's class, and all the questions are previous year questions. So with that being said, students, can I tell you a small story now? Students, can I tell you a small, very cutie story? Okay. So all of you know Gregor Mendel, and all of you know Gregor Mendel, our ultimate boss uncle, okay? Now, ultimate boss uncle, he did a lot of work in his lifetime. But the problem was, none of his work was recognized. Little sad, no? Little sad, because all the work he did not come into a proper light until 1900s. So more than 35 years or 30 years, his work was unrecognized and no one really appreciated his work. And that's a very sad story, and it's a very hard truth in science also. Sometimes you may work really hard, but until the right time or the destiny is there, all the work is not recognized. So with that being said, students, the same thing happened to Mendel. See, Mendel published his work of inheritance in the 1865. But until your 1900s, all the work was not recognized at all. But all of you know, the hard work never goes to vain. The hard work will always be fruitful some way or another, some time or other. And because of his work in the 1800s, we are still talking about Mendel. We are still recognizing his work, and his work is the foundation to the entire genetics. If there was no Mendel, probably we would not have the cure for all the diseases which we have right now, all the genetic disorders. So all the hard work pays some way or the other. If not at the right time, but someday it will definitely. And this is a proof of that. Okay. Now students, you need to understand why. There were several reasons, right? There were several reasons why Mendel's work was not recognized, right? There are several reasons why Mendel's work was not recognized. Let me tell you, there are 4 main reasons here. How many, students? There are 4 main reasons why Mendel's work was not recognized at that time. Now students, you tell me. Now if you want to talk to your friend, if you want to convey a message to your friend, how do you convey? For example, Sukanya wants to talk to Newton. Okay. How will she send the message? She'll open the WhatsApp, one tick, 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 tung, message sent. So it is very easy. Communication has become very easy and cheap right now. Right? It's very inexpensive. Back in my time, when I say back in my time, yes, there was a time there was no WhatsApp. And every single time I had to message a friend, I have to write a long paragraph and send one message. Why? Because every message used to cost me 1 rupee or 2 rupees. So very expensive back in the days. Okay. But right now messaging has become very cheap and very easy. Now students, imagine this was what I'm talking about is probably what, a decade old or almost 10 years back or 12 years back. But imagine Mendel's time. How difficult it would have been to convey messages or to publish something. Right now you have Instagram. You take one nice photo, if you make a painting, you'll click a picture, upload Instagram, you'll get immediate recognition. People will notice. But back in the days, noticing was very difficult. Publication was very difficult. Recognition was very difficult. So that was the first obstacle faced by the Mendel. That was the first obstacle faced by Mendel was what? That is communication. Communication was not very easy, students. The first main point was what? Communication was not very easy. The first main point, as it is now. Okay. Communication was not very easy. The first one. Secondly is that his concept of genes or factors. Now imagine, I am telling you, your mother is telling you every single day. Your mother is telling you every single day, you have to wake up very early. Waking up is very good. Okay, you will agree. But somewhere in the Instagram you will read, no, waking up early morning is not good. Sleeping late night is very good. Now you will go tell to your mom, Mummy, I read in the Instagram that waking up early is very bad. What will your mother do? She'll accept it, huh? Tell me, will she accept it nicely or one chappal will come at your face? Chappal will come at your face, no. So back in the day also, even though what I told you is not true, back in the day also same thing happened to Mendel. Okay. Whatever his new theories which he had, the theory of a gene or a factor or discrete units or blending theory, which is completely false. There is no blending of alleles, right? Law of dominance, law of segregation, law of independent assortment. So all the laws which he came up, right? All the laws which he came up with, everything was completely not acceptable by other professors or other colleagues or other scientists around the world. They're like, no, what is this nonsense? You go away, you go back to your village. Okay, that's what happened to Mendel. That is his concept of genes or factors in Mendel's words as stable and discrete units that control the expression of traits and the pair of alleles which did not blend. See, no blending with each other was not accepted by the contemporaries. So completely his theories was not accepted. Second point. First point was no proper communication. Second point is not accepted by the contemporaries as an explanation for apparent contribution to continuous variation seen in the nature. That was second one. The third one is very simple. Now tell me, all the bio students out there, tell me honestly, how many of you enjoy doing mathematics? All of you always associate maths as a separate entity and biology as a separate entity. Always we consider them separate, right? Biology is separate and maths is separate. Bio is interesting, maths little bit little headache. Okay. So back in the day also similar case was there. But let me tell you students, maths is very important in biology because right now, a fact, there are multiple mathematical equations which are being used to solve cancer, cure cancer. Like how a tumor grows, right? A tumor is growing. They're trying to use mathematics to cure cancer. So that is the importance of maths. So don't ignore maths, let me tell you. So back in Mendel's days also what happened? Back in the Mendel's days also, since Mendel was very interested in mathematics, statistics, he tried to include maths and statistics. He tried to include maths and statistics into the biology. Everyone were like, what is this? I cannot accept it. Not possible. All of you will agree with the same thing, right? You're like, maths is separate, biology is separate. So same thing happened to Mendel also. But that is one of the main reasons, that is, Mendel's approach using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists at the time. So third one was what? Third one was use of mathematics to approach in biology. That is the third one. There's one more main point now, students. The final point. What is the final point? The final though Mendel's work suggested that the factors or genes were discrete units, he could not provide any physical proof. There was no physical proof of existence of factors or any were made. Factors or say what they were made up of. So students, he could not prove that the existence of factors. Now can anyone in the chat tell me, anyone, think very hard, okay, think very hard. Why he could not prove the existence of genes or factors? Can anyone tell me? One simple answer is there. Only one simple answer. One simple answer, students. Anyone? Why was Mendel's, he could not prove the existence of factors or genes? The answer is very simple, students, because microscopy was not advanced. Only if you have proper microscopy, you can see the chromosomes, right? If there is no proper development of microscope, how would anyone notice the existence of chromosomes, or matter of fact genes also? So microscopy was not developed, so no one could prove that, Mendel could not prove that whatever genes or factors were actually in existence. Okay, it is like theoretical biology. Okay, so any doubt in this paragraph, students? Any doubt in this paragraph? Why Mendel's work was not accepted at that point? Okay, now as I told you, until 1900s, Mendel's work was not completely appreciated at all. But there came three saviors, the three main scientists who rediscovered Mendel's work. Students, three main scientists. The three main scientists who discovered Mendel's work was Ivory McLeod, that is De Vries, Carl Cons and Schmuck. Three main scientists, students, three main scientists who rediscovered Mendel's work, independently rediscovered Mendel's work, result on the inheritance of characters. Also, see, by this time, advancement in microscopy, advancement in microscopy at that time were taking place. Scientists were able to clearly observe cell division. So they were able to see cell division. Now cell division reminds me, can anyone in the chat tell me how many stages are there in cell division? Anyone? What are the three main stages of cell division? Can anyone tell me? The three main stages in cell division is G1, S, G2. G1 is G2. This is your interphase. This is your interphase. Okay, interphase. After that we have the M phase. Then we have the M phase or mitosis or meiosis phase. So all of this was clearly visible now. People were able to hypothesize, scientists were able to tell the cell division properly. Now students, if you remember cell division, if you have studied cell division, especially meiosis, in meiosis, can we clearly tell the recombination is happening at your prophase 1? Can we clearly tell there is in metaphase chromosomes are aligning on the equatorial plate? We can tell. And the anaphase chromosomes are separating. In the case of meiosis 1, homologous chromosomes separating. And telophase, what is happening? Entire thing is rearranging and we are getting two cells. Right? All the chromatin or chromosomes separating out. So with the help of microscopy, we were able to see the movement of chromosomes. Yes or no? So here also the scientists were able to see the chromosome movement also. Okay, see here. This led to the discovery of structures in the nucleus that appear to be double and divide. See, double and divide, appear to be double and divide just before each cell division. These were called as chromosomes. They were called as what? Chromosomes. So finally, into the timeline, chromosome is entering now. Now we can see chromosomes. We can see division of chromosomes. Yes or no? We can see division of chromosomes right now. Right? By 1902, just 2 years after that, the chromosome movement during meiosis, had been worked out. Finally, in the 1902, we can see the movement of chromosomes. How the chromosomes are moving in meiosis. What is happening in prophase, metaphase, anaphase, telophase. What is happening in cytokinesis. Everything had been worked out by 1902. Okay. Now students, here two more scientists comes. That is Certain and Boveri. Certain and Boveri noted that the behavior of chromosome, the way the chromosome is moving, similarly even the gene is moving. I'll give you a quick example, understand. Now imagine this is an entire chromosome. This is an entire chromosome. Okay. Now in this hand, do I have fingers? Yes sir, I have fingers on my hand. Similarly, on a chromosome, do we have genes? Yes. So where are technically genes present? Genes are present on the chromosome. All of you agree with me? Genes are present on the chromosome, just like how on this hand I have fingers. Now you tell me students, if I am moving my hand from here to here, am I also moving my fingers along with it? Yes, I am moving. If I am moving chromosome from here to here, are the genes also moving along with it? Yes. So can I tell if chromosome is moving, even the genes are moving? Yes. Can I tell if chromosomes are separating? Can I tell even the alleles are separating? Yes. Why? Because as we know, the homologous pair of chromosome, one chromosome is coming from mother, one chromosome is coming from father. In the two chromosomes, one is from mother, one is from father. Now similarly, one allele is coming from mother and one allele is coming from father. So two alleles, one pair of chromosome, one pair of chromosome. So if the chromosomes are moving, even the genes are moving. The behavior of chromosome is very similar to behavior of a gene. Why? Because gene is present on the chromosome. If the chromosome is moving, even the genes are moving. That is the meaning of chromosomal theory of inheritance. Chromosomal theory of inheritance says that there is a relation, there is a parallelism between the behavior of a chromosome and a gene. That's all, students, that's all. See here. Certain and Bouvier noted that the behavior of chromosome was parallel to the behavior of a gene, and use the movement of chromosome during spermatogenesis, right? Movement of chromosome to explain the Mendel's law. Recall, if you have studied in the behavior of chromosome during mitosis, we have seen, in meiosis we have seen. The important thing to remember that is the chromosomes as well as genes. Chromosomes as well as genes occur in pair. Chromosome occur in pair, genes occur in pair. There are one chromosome, there are two chromosomes, there are two alleles. Right? The two alleles, very important line. The two alleles of a gene pair, are located on the homologous site on the homologous chromosome. What is the meaning of that, students? If you look at this, this is one pair of homologous chromosome. In this pair of homologous chromosomes, here we have one allele, here we have one more allele, and both are on a homologous site. The meaning of homologous site is also called as loci, that is a fixed site. So if I can see on this homologous chromosome, if I can see in this homologous chromosome, we can find exactly one gene here and one gene here. Yes or no? So in this chromatid, one allele is coming from here, one allele is coming from here, one from mother, one from father, one from mother, one from father. Clear? So that is the meaning of the last line. The meaning of last line is that the two alleles of a gene pair, okay, two alleles of a gene pair are located on a homologous site. This is called as homologous site. Homologous site on a homologous chromosome. Sir, please explain traits, allele, gene once. First part. What's the first part? I have explained beautifully there. What is the difference between a character and a trait? What is a gene? What is a allele? What is a homologous chromosome? What is a heterozygous chromosome? So what's the first part? You'll get a better idea on that. Okay. So students, here also we can see this is basically your meiosis, right? This is meiosis. Here we have G1, S. Here we have one chromatid. The chromatid will become double in the S phase. Then we have G2. Can you see? Then there is something called as meiosis anaphase 1. Can anyone tell me what exactly happens in anaphase? Is there separation of homologous chromosome or is there separation of sister chromatids? Tell me in the chat quickly. In the case of your meiosis 1 anaphase, there is separation of homologous chromosomes are separating out. See, one pair, one more pair. Two homologous chromosomes separated. And this is at meiosis 2, what happens? At meiosis 2, there is separation of sister chromatids. At meiosis 2, separation of sister chromatid. Here, separation of homologous chromosome. Here, separation of sister chromatids. Separation of sister chromatid. Finally, we have the 4 germ cell. So, chromosomal theory of inheritance. Chromosomal theory of inheritance says one thing, that is, behavior of a gene is similar to behavior of a chromosome. If chromosome is moving, gene also is moving. Now imagine all of you, All of you are gene. You are sitting in a car. If the car is moving, will you also move with the car? If you are sitting in the car, obviously. If you are sitting in the car, if the car is moving, you are also moving. So imagine the car is the chromosome, and gene is present on the chromosome. So if the chromosome is moving, then the gene also is moving. So their behavior is same to same. Behavior is same to same. I believe the concept you are able to understand. You don't have to by heart all this. Okay. Now here they have given you comparison. One is a chromosome, one is a gene. Both of them have very much similarities. Can you see? Similarities is there. Segregation at the time of gamete formation such that only one of the pair is transmitted to a gamete. One of the pair is this is a gamete. This is a gamete. One of the pair is transferred to a gamete. Yes or no? Can you see here? One of the pair is transferred to a gamete. Yes, sir. In the case of your genes also, capital T, small t, one of the pair is transferred to each gamete. Yes or no? Can you see here? Segregation at gamete formation, and only one of each pair transmitted to a gamete. Law of segregation. Independent pair segregate independent of each other. That is law of independent assortment. This is your gene. One pair segregates independently of the other pair. Law of independent assortment. Okay. Here again the same example is given what we learned before. Same example is given here also with two possibilities. In the first possibility we have orange and green. Here we have orange and red. Same thing. Anaphase, separation of homologous chromosomes, and then we have your germ cell. Finally, each one is going to a gamete. Okay, anaphase 2, separation of sister chromatids, separation of homologous chromosome. Okay, so students, that was your homologous chromosomes and your entire theory of chromosomal theory of inheritance. But there came a scientist. Who gave a physical proof. He gave a physical proof that this is actually happening and it is not a random theory. Okay, so the physical proof was given by the T.H. Morgan. Same T.H. Morgan who worked on the Drosophila melanogaster, the fruit fly. Okay, so the Surton and Boveri argued that the pairing and separation of pair of chromosomes, would lead to the segregation of pair of factors they carried. So obviously, if the chromosomes are separating, even the factors will also separate, alleles will also separate. Okay, certain united the knowledge of chromosomal theory of inheritance plus Mendelian principles, two principles together. Finally, we got the chromosomal theory of inheritance. That is the end of it. All of you understood? Tell me yes or no in the chat. Students, quickly. Students, if you do not show the josh or the energy or the, coming to the live and everything, it gets a little demotivated, right? We are disappointed. So if you are not calling your student friends, if you're not liking the video, if you're not commenting, there's no point in taking on YouTube, right? So see, we have been telling you with the Tejas series also. Tejas series, if there's a decrease in the energy from your side, we will stop Tejas series next week only. I'm telling now only. Okay. Now students, following this synthesis of idea, experimental verification of the chromosomal theory of inheritance was given by T.H. Morgan, Thomas Hunt Morgan. Such a cool name. Hunt is there in the name only. And his colleagues led to the discovery of basis of the variation at sexual reproduction produced. Morgan worked on the Drosophila melanogaster. Now students, if you remember, we learned in the pea plant, right? In the case of pea plant, we learned why the pea plant by Mendel. Similarly, now also we will learn why the Drosophila by the T.H. Morgan. Why Drosophila only? See here students, one by one I'll make you the point. See, which were found very suitable for such studies. First point what? The first point is that they could be grown on the simple synthetic media in a laboratory. So easy growth, first point. Second point is that they complete their life cycle. Second point is they complete their life cycle in about 2 weeks. Done. Third, a single mating could produce a large number of progenies. Third one. Fourth one is what? Fourth one is that there is a clear differentiation about the sexes, male and female. Male Drosophila, female Drosophila could easily be identified. Okay, so those were the four main reasons why your T.H. Morgan chose the fruit flies. Now students, you might ask me, sir, how do I capture the fruit fly? Very simple. Eat a banana, throw the peel somewhere. On the peel, within 10 minutes, you can see the fruit flies on the peel. Okay, very easy to capture also. Okay. Now we come to the concept of linkage and recombination. Now students, I need 2 volunteers from the chat. 2 volunteers who wants to volunteer for the explanation. Anyone? 2 volunteers. 2 volunteers from the chat, all of you. So Morgan carried out several hybridization experiments. Morgan carried out several dihybrid crosses, just like Mendel, in Drosophila to study the genes were sex linked. Okay, so all the genes were sex linked. Okay, their crosses were similar to a dihybrid cross carried out by Mendel in pea plants. Okay, these were much more similar. For example, Morgan hybridized yellow body. He crossed yellow bodied white eye crossing with brown body red eye. Now students, you need to tell me, is this monohybrid cross or the dihybrid cross? Okay. Tell me students, anyone, tell me, is this monohybrid cross or is it dihybrid cross? Since we have taken 2 different characters here, one is your body color, one is eye color. So can I call it technically a type of dihybrid cross? Yes sir, dihybrid cross. Okay, dihybrid cross. And intercross their F1 progenies. So when we cross, we get F1. Then there is selfing of F1 into F1. So it is surprising. Now can anyone remind me, what was the phenotypic ratio? What was the phenotypic ratio of dihybrid cross in the case of Mendel's experiment? In the case of Graham Mendel's experiment, the dihybrid cross for the phenotype was 9:3:3:1. Yes or no? 9:3:3:1 was the phenotypic ratio of the dihybrid cross. But, what is the but here? When T.H. Morgan did the same experiment, same dihybrid experiment, the ratio which he obtained was not at all 9:3:3:1. It was not the ratio which he expected. This ratio was not obtained. This ratio was not obtained at all. It's like, what is this? Mendel is telling me we are getting 9:3:3:1 ratio, 9:3:3:1 ratio. But when I'm doing the experiment, I'm getting different result. Now, what is the mystery behind it? Why am I getting a different kind of ratio compared to Mendel? Interesting, no? Then same thing happened to T.H. Morgan also. So he decided to look into more detail of it, and later on he identified that. Students, later on he identified that line is very important for you to understand. Later on he found that, students, he quickly, when the 2 genes in a dihybrid cross, okay, the 2 genes in a dihybrid cross were situated on the same chromosome. This is a very important line. So students, so when Mendel did the cross, with the characters which he took, the Mendel's experiment, all the characters were present on different chromosomes, 4 different chromosomes to be exact. The characters taken by Mendel was on 4 different chromosomes. If the characters are present on 4 different chromosomes, there was some room for recombination or crossing over. But when the T.H. Morgan did the experiment. When T.H. Morgan did the experiment, the characters were present on the same chromosome and there was no crossing over at all. Now that gave us the idea about linkage and recombination. It gave us idea about linkage and recombination. Now what is linkage and what is recombination? Let's try to understand in a very simple terms. Okay. Okay, let's take Pooja and Sneha. Okay, let's take Pooja and Sneha first of all here. So here I have Pooja. Okay. Here I have Pooja here. Here we have Pooja. I don't know why one eye is. Okay, this is Pooja. Okay, here we have Pooja. Okay, done. Now here we have one more girl, Sukanya. Okay. Okay, we need one more person. We need one more person today. We need one more person. Let's take Mardel. Okay, Mardel will take. I'm sorry if I'm pronouncing your name wrong. I don't know. I'll just take two girls. Some random girl. Don't worry. This is one more random girl. Okay. Now tell me, students, these two girls. Okay, here is the name only. Let's call it girl A, girl B. Girl A, girl B, and girl C. Okay. No complexities. Okay. Now, students, between them, these two girls are best friends. These two girls are best friends. They'll hold hands and go. Okay, we'll hold. Make them hold their hands also. Okay. These two girls are best friends. Okay. These two girls are best friends. Now tell me, students, does these two best friends, between them, will the bond will be stronger or very weak? Tell me. Very close friends, childhood friends they are. Childhood friends. Between them, do you think the bond will be very strong or will they not have very weak bond? Obviously, students, between these two friends, can I tell the bond will be very strong? So can I tell instead of bond, linkage between them will be very strong. Okay. Linkage will be very strong. Okay, very strong linkage. If linkage is very strong between these two people, will they fight with each other? Will they show dushum? Obviously not. There is no cat fight there. So between A and B, can I tell if fighting is very less, can I call it recombination? Recombination is going to be very less. Fighting will be very less. Okay, fighting will be very less. Okay. Now, these two girls are not very friendly. Okay, these two girls are not very friendly. They do not get along. When Cold War is going on, they do not like each other. Okay, they do not like each other. Now tell me, students, they will have a strong bond or a weak bond? They're like, what bond? No bond at all. But technically, there's no bond at all, right? So they will have very weak bond between them. So can I tell between A and C, the bond is very less. If the bond is very less, students, will they fight with each other? Obviously, when Cold War is going on. So can I tell recombination is very less. Recombination is very high. Fighting is very high. Okay, fighting is very high. Done. Now, students, I will give you perspective now. Understand? For example, this is a chromosome. On this chromosome right here, we have gene A, we have gene B, and we have gene C here. Now tell me, students, between gene A and gene B, they will have very high linkage. So can I tell linkage between them is very high? Or can I say the genes are located very close to each other? So can I tell you, students, gene A and gene B are very close to each other? So can I say they are tightly bound to each other? Yes, tightly bound to each other. So they are very close to each other and tightly bound to each other. Can I also say that their linkage is very high? And recombination is very low. Between gene A and gene C. Now, students, you tell me, what about gene A and gene C now, between these two people, here, gene A, gene C, gene A and gene C, between gene A and gene C now, between gene A and gene C, can I say they're very far from each other? The two genes are located very far from each other. Yes, sir, they're very far from each other. So can I also tell between gene A and gene C, they're very loosely bound? Yes or no? They're very loosely bound to each other. Okay, they're very loosely bound to each other. Very true. So can I tell at the same time linkage is very low here, recombination is very high. Recombination is very high. Now students, understand the perspective now. What is actually happening here? Your linkage is actually inversely proportional to recombination. If linkage is high, recombination will be low. If recombination is high, linkage will be very low. So it is completely inversely proportional to each other. Now students, why did I teach you this now? The reason is very simple. That when the T.H. Morgan did the experiment, on Drosophila melanogaster, he noticed that the 9:3:3 ratio was not obtained. The reason why 9:3:3:1 ratio was not obtained is because the two genes which was present in his experiment, which was giving the characters, that is body color and eye color, the body color and eye color genes were located on the same chromosome very close to each other. If they are present on the same chromosome very close to each other, will there be recombination? You tell me now students. Will there be recombination? Yes or no, tell me. Here, imagine these two genes are located very close to each other. These two genes are located very close to each other. This is the chromosome. On this chromosome here we have gene A and we have gene B, gene A and gene B. If these two genes are located on the chromosome which are very close to each other, obviously there will be no variation. Obviously there will be very less variation, and we will not get especially these two ones, this 3 and this 3. These two variations would not be obtained easily. Why? Because in the 9:3:1 ratio, 9 is parental, 1 is parental, 3 and 3 are what? 3 and 3 are recombinant. That is not obtained. Okay, see here. It's given here. He observed that the two genes did not segregate independently. Why? Because the two genes are present together. The two genes are present together on the same chromosome. Okay, of each other, and the F2 ratio deviated from the very significantly from 9:3:3:1 ratio expected when the two genes are independent. So technically speaking, he was disproving the law of independent assortment to some extent. Okay, so the two genes will segregate independently when they are very far from each other or they are present in different chromosome. If the two genes are located very close, they will not assort independently. They will go together. They will be going together. They will not be going separate ways. I hope that concept you are able to understand, because this is a very conceptual, technical understanding. You can't mug all this up. If you just read the lines of NCERT, it becomes very difficult to understand the meaning of linkage and recombination. And with the help of linkage and recombination, how your T.H. Morgan got a deviation from 9:3:3:1 ratio. Done? Yes or no? All of you understand the concept today? Smash the like button, all of you. See here. Here we have, see, two genes are linked here on the same chromosome. The two genes are very close to each other. Linkage is very high, recombination is very low. Linkage is very high here. The linkage is very high here. In this case, the two genes are completely not linked. Linkage is very low here. In this case also, two genes are on the same chromosome, but they're far from each other. Again, linkage can be little low. Understanding? The same diagram is also given in your NCERT. Now, what is in the case of Mendel? In the case of Mendel, what happened? In the case of Mendel, the two genes were present on different chromosomes. 4 different chromosomes actually. Okay. See here. This is called as recombination, all of you know. Called as recombination, all of you know. Okay. Now, friends, Morgan and his group knew that genes were located on the X chromosome. Very important line. All the genes were located on the X chromosome, and saw quickly that the two genes in a dihybrid cross were situated on same chromosome. The proportion of parental gene combination which were much more higher than the non-parental type. The Morgan attributed due to the physical association or linkage of two genes, and the coined term linkage. So what is linkage, students? Linkage is very simple. What is linkage? Linkage is nothing but physical association of two genes on a chromosome. I'll repeat again. Physical association of two genes on a chromosome is called as linkage. It is called as linkage. Then what is recombination? Recombination is generation of non-parental type. Okay. This physical association of genes on a chromosome and recombination to describe generation of non-parental gene combination. Non-parental gene combination. Okay. Non-parental gene combination. Here they have told you about something called as tightly linked. See, meaning is what? Linkage is very high. Recombination is very low. Recombination is very low. 1.3 recombination. Wide in miniature, Wing showed recombination is very high. So linkage is low here. See here, students. NCERT will show you. In this diagram, can you see? In this diagram, you will be able to notice your Y and W. Y and W, they are very close to each other. The two genes are very close to each other here. Yes or no? The two genes are very close to each other. So can I tell when the two genes are very close to each other, recombination is very low. Why? Linkage is very high. Tightly bound, linkage is high. Recombination is low. See, recombination is very, very low. Linkage is very high. Parental is high. At the same time, if you notice this one, the body color and wing size, two genes are very far from each other. Yes or no? Can you see here? The two genes are far from each other. If you notice here, the two genes are far from each other here. So if the two genes are very far from each other, so can I tell linkage is low here? In this case, linkage is low here. Linkage is low and recombination is high. Recombination is higher. Can you see? Recombination is 37.2. 37.2. Done. That's all. Okay. Now students. Now one more scientist there. His student. Whose student? T.H. Morgan student Alfred Stewart Went used the frequency of recombination, that is how often the recombination is happening, frequency of recombination between gene pair on the same chromosome. How often the recombination is happening on the same chromosome. How often the recombination is happening on the same chromosome, okay, or the gene on the same chromosome as a measure of distance. He called it distance between the genes and called it as mapped their position on the chromosome. And today's genetics are extensively used as the starting point in the sequencing. Basically, Alfred Stewart Went he founded a new value, SI unit. All of you call SI unit, right? 1 kilometer, 1 meter, centimeter, millimeter. Similar to all these SI units, Alfred Stewart Went introduced a new unit. He introduced a new unit and we call it centimorgan or map units. It is also called as map units. What is map units, students? What is map units? Map units is nothing but distance between the two genes on a chromosome. Distance between two genes on a chromosome is called as a map unit. This is 9 map units, 9.5 map units. Okay, map units. Now let's see how many of you are able to solve this question. Very simple question on the board. Go. Time starts now, all of you. This right here is a NEET 2023 question. Last year. The frequency of recombination between the gene pair on the same chromosome as a measure of the distance between the genes to map the position on the chromosome was used by. Basically, who discovered the map units? Map unit or centimorgan? Map units or centimorgan? Who discovered that? Who discovered that? All of you know Alfred Stewart. Next question. The chromosomal theory of inheritance was proposed by. NEET 2022 question. NEET 2022 question. Who discovered the chromosomal theory of inheritance? Very simple question. Certain and Boveri. Yes, Certain and Boveri. Next question. Mendel's law of independent assortment does not hold good. See, I told you, it does not hold good at all. When genes that are located close on the same chromosome. I told you, when T.H. Morgan did the experiment, when the chromosomes are present, when the genes are present on the same chromosome, chromosomal theory of inheritance does not hold good. True. Assertion is correct. Closely associated genes assort independently, students. When the two genes are very close to each other, do they separate out or they go together? Tell me. When the two genes are linked tightly, linkage is very high, do they separate out easily? Do they show recombination? Obviously not. So can I tell it is wrong? Wrong. So what is the answer? A is true, R is false. NEET 2022 question. Okay. Next question. In Drosophila, the genes for color of the body and the color of the eye are situated on both the sex chromosomes, autosomes, Y chromosome or X chromosome. Line of NCERT, students, line of NCERT, direct line of NCERT, the question is there. All of you know, Sir T.H. Morgan and his colleagues worked on the X chromosome. It was the X chromosome. NEET 2022 question again. X chromosome. Now, a low frequency recombination indicates that genes are located in. Basically, low recombination means what, students? A low recombination means very high linkage. Yes or no? Very high linkage. The meaning of very high linkage is what? The meaning of very high linkage is the two genes on the chromosome are very close to each other. Means we're very tightly bound. Yes. Close to each other. NEET 2022 question again. So students, now do you get an idea? How the questions can be asked to you? Similarly, there are many questions in today's class. Many questions are there. So one by one, let's tackle every question and we go ahead. Okay. So all of you smash the like button because many questions are coming up, especially for the diseases and the syndromes. Many questions are there. So stay tuned till the very end to solve all the questions. Now the next question, or the next concept. The next concept is called as polygenetic inheritance. Now students, in Mendel's law of dominance or law of segregation or law of independent assortment, did we ever talk about intermediate plant height, or did we always consider a tall plant or dwarf plant? Tell me in the chat, all of you know. Tell me. Did we always consider a very tall plant or dwarf plant, or did we have intermediate anywhere intermediate? We always have violet flower and white flower. Did we have something in the middle in Mendel's? No. See, incomplete dominance, yes we had, but that is not Mendel's. That is not Mendel's work. Okay. Similarly, in the case of Mendel, did we ever find some pink color seed? No. We always had a green seed and a yellow seed color. Round seed, wrinkled seed. So can I tell, in the case of Mendel's work, we always had two extreme ends. One tall, one short. One tall, one dwarf. We never had medium height. Now all of you look around. In your classmates, do you have always only tall people, only short people? One is 6 feet, everyone else is 4 feet. Not possible, no. In the case of humans, do you notice that there is 5 feet, 5'1", 5'2", 5'3", 5'4", 5'5", 5'6", 5'7", 5'8", 5'9", 5'10", 5'11", and 6 feet, 4'1", 4'2", 4'3". So in the case of human height, we have something called as intermediate steps. Yes or no? Yes, sir. In the case of human skin color, we have extreme dark, extreme light, but we also have intermediate colors, light brown, little bit fair, less fair, little bit darkness, more darkness. So we have a gradation in skin color also. Yes, sir. So all of this phenomena cannot be explained with the help of Mendel's work. Cannot be explained, no, not possible at all. So all of this work can be explained with the help of something called as polygenetic inheritance. So polygenetic inheritance explains all the intermediate traits. See here, purple and white. But if you look around, you will find there are many traits which are not so distinct in their occurrence or spread across the gradient. For example, humans, we just don't have tall or short people, we have intermediate. The same goes for your skin color. Same goes for skin color. So what exactly is polygenetic inheritance? What exactly is, students, polygenetic inheritance? In the case of polygenic inheritance, many genes are responsible for one trait. Many genes are responsible for one trait in the case of polygenetic inheritance. Many genes, one trait. Okay, it's given here. See here. Alternately, but whole range of possible height. Such traits are generally controlled by 3 or more genes and thus called as polygenetic inheritance. Now students, you tell me, there is a very good chance that you will be very tall in life. Mother is tall, father is tall, very tall. But you happen to have no nutrition at all. You are not getting any nutrition in your diet to grow tall. In that case also, will you grow tall? Tell me. You're not getting a proper protein, you're not getting proper carbohydrates, you're not drinking milk regularly. Will you grow tall still? No. There's a very high chance that your parents may be tall, but you will not be tall because of no proper nutrition. Yes or no? There's a very high chance that your father is very light color, your mother is also very light color, but you're dancing in the sun whole day. There's a very high chance that you'll get tanned. You get tanned, you look dark. Yes, that is also good anyway, but all I'm saying is, all of this polygenetic inheritance is environmentally controlled. Environment also plays a very crucial role in the inheritance of these genes or expression of these genes, not inheritance, expression of these genes. Okay. Besides involvement of multiple genes, polygenic inheritance, it can also take into account, that is, influence of environment. Human skin color is another example of the polygenic inheritance. Polygenic inheritance phenotype reflects the classic example of this. The polygenic inheritance or trait, the phenotype reflects the contribution to each allele. The effect of each allele is additive. Now what is the meaning of that? Here we can see something called as additive effect. Now what is the meaning of additive effect, students? Understand. For example, every single allele plays a very important role here. No. Phenylketonuria we will study in the case of pleiotropy, not here. Okay. See, what is the meaning of additive effect? Understand additive effect. In the case of additive effect, when all the dominant alleles are present together, the skin color will be very dark skin color. Okay. But in the same case, when all the recessive alleles are present together, small a, small b, small b, small c, in such cases, the skin color is very light skin color, okay. So when I say additive effect, the more number of dominant alleles, expression is stronger. What I mean by that? In this case, now we have capital A, capital B, small c, small c. Now here we only have 3 dominant alleles and 3 recessive alleles. This can lead to brown color. Now this is the lightest color. Now here, what if, now in the next case we have capital A, small b, small c, small c. In such cases, only one dominant allele is present. In such cases, the skin color will be just light. Here we have lightest, all recessive alleles. So understand, the meaning here is, the more number of dominant alleles, the effect is going to be stronger, and the effect is going to be what? Additive in nature. Additive effect. It is called as additive effect. The stronger, the more the alleles, stronger is the phenotypic expression. So many genes, one gene, second gene, third gene, many genes are controlling one phenotype. That is called as polygenic inheritance, and it is also additive in nature. Okay, see here, dark light, moonlight is here. Okay, see here, the genotype with dominant allele will have the darkest color. At the same time, lightest color here. Okay, now students, as expected, the genotype will be 3 dominant alleles, recessive alleles. Okay, all of this will be intermediate. In this manner, the number of each allele, each type of allele in the genotype would determine the darkness or lightness of the skin in the individual. That is called as your, what is that? That is called as polygenetic inheritance. Polygenetic inheritance mainly determines the number of alleles are present, stronger the effect. Okay. Now Malia was asking me, what is, sir, where do you study? Sir, where do we study the phenylketonuria, students? Phenylketonuria. Understand? In the case of polygenetic inheritance, many genes, one trait. But in the case of pleiotropy, single gene, many traits. Here. So it is completely opposite. In the case of your pleiotropy. A single gene, many traits. In the case of polygenic, many genes, one trait. So here we have gene A, gene B, gene C, all of them for one trait here. One trait. Many genes, one trait in the case of polygenic inheritance. But in the case of pleiotropy, a single gene, multiple phenotypic expression. Trait A, trait B, trait C. Understood, all of you? So it is completely opposite. That is pleiotropy. Now let's read what is given in NCERT. Okay. Students, I have always told you, this is like a mother. Okay. Mother can, always do all the work in the house. Surprise. I don't know how come they do it. My mom, she will cook also. She will, work also. She will, obviously give me work also. So how your one mother can do all the functions in the house, multi-talented. Okay. Similarly, one gene is multi-talented. One single gene can show multiple phenotypic expression or multiple traits. Okay. Now see here. There is, however, instances where a single gene, see it's given here, a single gene is called as pleiotropic gene. Okay. The underlying mechanism of pleiotropy in most cases is the effect of a gene on a metabolic pathway. Okay. So a gene will affect the metabolic pathway, and because of that change in metabolic pathway, will lead to different types of effect. Okay, now what is the metabolic pathways here? For example, disease called as phenylketonuria. Okay, disease is here, which occurs in the humans. Okay, it is occurring in the human. Now in this disease, what is happening is there is something called as a point mutation. There is a mutation here. There is a mutation in a gene, and because of the mutation, there is one enzyme called as phenylalanine hydroxylase. And if this enzyme is not being produced, it can have multiple expressions. See here. This disease is caused by a mutation in a gene that codes for an enzyme called as phenylalanine hydroxylase. A single gene mutation, this manifests itself through phenotypic expression. What are the causes? What are symptoms here? The symptoms, three main symptoms. The first symptom is mental retardedness, reduction in hair and skin pigmentation. So change in one gene can show multiple phenotypic expression. Understood all of you? So there is a major difference between your pleiotropy as well as your polygenetic inheritance. In pleiotropy, a single gene, many expressions. Solve this question, students. Solve this question. See, in examination, they will ask you a direct question and you will read it in English itself. See here. NEET 2024 question. NEET 2024 question. This year, NEET examination questions. Let's see how many of you will be able to solve this question. The phenomena where a single gene exhibits multiple phenotypic expression is called as pleiotropy. The students yes, we will get to the exam. See students, I'll teach you what exactly happens in phenylketonuria. In phenylketonuria, phenylalanine is not getting converted to tyrosine. We'll come to the disease one by one. Now what is multiple allelism, students? Don't get confused. Multiple allelism is that one gene will have multiple alleles. That is called as multiple allelism. It is completely different. Okay, completely different. Now students, the phenomena of pleiotropism refers to, what is the meaning of pleiotropism? More than two genes affecting a single character. Two genes? Not really. Presence of several alleles for a single gene controlling a single crossover. Presence of two alleles, each of the two genes controlling a single trait. No. A single gene affecting multiple phenotypic expressions. Yes or no? NEET 2023 question. So easy answer. Easy question. Easy answer. A single gene, multiple phenotypic expressions. Yes or no? Done. Next question. Select the correct statement with respect to pleiotropy. Students, do you realize from NEET 2022, 23, 24, all the 3 years, one question has come from pleiotropy. A gene is said to be pleiotropic if it affects more than one trait, right? It is affecting more than one trait. Phenylketonuria is an example of pleiotropy. Yes, it is an example of pleiotropy. A condition where one gene has several alleles is referred to as pleiotropy. No, this is multiple allelism. A trait is said to be pleiotropic if several genes are controlling it. No, not several genes, only one gene. So what is the answer? A and B. What is the answer? A and B. Okay, these two are right answers. Understood? Again students, NEET 2022 again. NEET 2022 question. Read the question. Okay. Assuming fur color of an animal is dark, range of color, shade and white. Okay. Range of color, shade and white. A cross is made between capital A, capital B, capital C, capital C. That is dark fur color and female with small a, small b, small b, small c with white fur color. So dark fur color, white fur color, multiple genes controlling one phenotypic expression, fur color. What does it remind you of? Many genes, one trait. What would be the color of the F1 generation? All intermediate color, range of color shades, all dark color, all white color. Students, in the case of pleiotropism, this was not the case. Pleiotropism, one gene, multiple expression. But right now, multiple expression, one gene. That is a typical example of polygenetic inheritance, where we get intermediate, yes or no? So all intermediate color, Now students, that is the end of the two main concepts done and dusted. Now we will go to the one more concept called as sex determination, all of you. Okay. Now students, if you want all of these notes are available on the Telegram channel, all of you. Basavaraj Sir Biology. If you join the Telegram channel, all the notes are available here. You can download them and write them and learn them on your own. Okay. So let's go to the next concept now. Okay, can we start? Students? That is called as sex determination. Very simple, very easy to understand. But before we understand the entire sex determination, there is a small concept as the scientist who first became a pioneer in this field, or the first scientist who is like, this is this. Who is a scientist? So it's very important to remember a scientist. It's very important to remember a scientist, and his name was Henking. Name only as King. Okay. The initial clue about genetics and chromosome mechanism of sex determination can be traced back to some experimental carried out on insects. Obviously, you cannot carry out a random experiment on humans. Very difficult. So the first sex determination experiments was carried out on insects. Okay. In fact, the cytological observations made in the number of insects led back to the development of concept of genetics and chromosomal base of sex determination. So students, you need to understand here is, there was a scientist called as Henking. Very important scientist, students. Very important scientist called as Henking. Now this scientist noticed that after spermatogenesis in the case of insects, 50% of sperm, he noticed that 50% of sperm had an nuclear material. A special nuclear material was present. He noticed that in the 50% of sperm there was a nuclear material. Okay. He also noticed that in other 50% of sperm, the nuclear material was completely missing. It was completely missing here. Okay, it was completely missing, or it did not receive the nuclear material. So back then he could not identify. He was not able to identify what was this missing nuclear material, right? So all he did was, since he is not able to find what is this missing, always right, in biology or in mathematics, if something is missing, or if you have to find something but you don't know what it is, what do you call that? X, right? X. So here he also called it as X body. Now students, later on, after all this many years, we found that the scientist Henking, which he referred to as X body, it was none other than the X chromosome. When today we know it as, it is nothing but X chromosome. Okay, X chromosome. Now see, Henking in the year 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas other 50% sperm did not receive. Henking gave the structure as X body, but could not explain significance. Further investigation by other scientists led to the conclusion that the X body, the Henking was in fact the X chromosome. It was in fact the X chromosome. It was the X chromosome. Now students, I'll make you write in a very easy manner the entire sex determination. So all of you open your books. Write down and remember this. Okay. Now what exactly did he do? He found something called as male heterogamety. After Henking, what is sex determination? Students? There is something called as male heterogamety. Male heterogamety. Now, what exactly happens in the case of male heterogamety? In the case of male heterogamety, the males decide the sex of the offspring. Will the offspring or the child or the progeny, will he be male or female? That is decided by the males. Why? Because the males here will have two different types of sex chromosomes. Okay. For example, in the case of humans, tell me students, in the case of humans, which two chromosomes do we have, students? In the case of humans and Drosophila also. In the case of humans and Drosophila, we usually find XX and XY. Yes or no? XX and XY. Here XY is usually the male and XX is usually the female. It is always the female. Not usually. It is always the female. And at the same time, the XX will XY will determine the sex of the child. That is why it's called as what? That is why it's called as male heterogamety. Hetero means different, gamete means gametes. The males have two different gametes here. And it is always the males who decide the sex of the child. Okay. Now, other example is grasshopper. The other example is going to be grasshopper. In the case of grasshopper, there is something called as XX, XX and XO. XX and XO. Here XX is again female and XO is always the male. And it is the same male who determines the sex of the offspring in the case of grasshopper. But you need to see a small difference here. The small difference here is that in the case of males, both of them have 46 chromosomes. But in the case of grasshopper, the males will have what? The males will have one less chromosome. Okay, this is XO. XO means one is missing. In the case of males, one will be missing. That is called as male heterogamety. The second one is called as female heterogamety. It's called as what? Female heterogamety. Now again, in the case of female heterogamety, it is the females who decide the sex of the offspring. The first example is in the case of birds. Birds or your hen, chicken, everything. In the case of birds. The first example is in the case of bird, students. Okay, in the case of birds. Now, in the case of birds, what is happening here is the chromosome in the case of female is different now. Understand. Okay. Here we have ZW and ZZ. Here the ZW is the female, ZZ is the male here. And the sex of the offspring is determined by the female here. The female decides the sex of the child. Okay, that is the birds. In the case of butterfly, we have ZZ and ZO. In the case of butterfly, we have the ZZ, ZZ and ZO. Here we have this is the female and this is the male again. Again, the sex is determined by the female. That is male heterogamety and female heterogamety. Quickly tell me, students, any doubt till now? Male and female heterogamety. Very easy, right? Very easy. Okay. The same thing is given everywhere. Okay, you can read the entire thing and that's the same thing. Here we have 6 chromosomes, all of you know, and we have autosomes, the normal chromosomes, autosomes. Okay. It is given here also, you can see. Okay. See, same thing is given everywhere. You can have a read at it. Okay. Male and female, male heterogamety, female heterogamety. Students, all of you know, right? In the case of humans, how does it happen? In the case of humans, it's very simple, right? In the case of humans, all of you are aware. In the case of, one second. In the case of humans, what is happening? In the case of humans, we have male and female. Males usually have what? Males usually have the X and Y. Female have the XX chromosome. When two X chromosomes meet together, we get XX. XY, we get male here. And here we have usually have the female child. Here we have male child. Here we have the male child. This is the sex determination in the case of humans. Male heterogamety, female heterogamety. Done. Sex determination in humans. Just not did that. Now, students, the next thing is going to be the sex determination in the case of honeybee. Very, very amazing it is. It is called as haplodiploidy. It is called as haplodiploidy. Let me tell you why. In the case of honeybee. In the case of honeybee. Understand. In the case of honeybee, it's called as haplodiploidy. Haplodiploidy. So what exactly happens in the case of haplodiploidy, students? Understand the first main point is very important. In the case of honeybees, this happens in the case of honeybees. Honeybees, ants and wasp. Wasp also. Honeybees, ants and wasp. Ants and wasp. Okay. Now what exactly happens here? Here the sex is defined by sex of the offspring is based on, it is based on number of set of chromosome. Number of set of chromosome. That is very simple. If the organism here, if it is male, if he is male, he will only be haploid, while the female will be diploid here. The female will be diploid here. So the number of set of chromosomes, the number of set of chromosomes determine the sex of the child. If haploid set of chromosomes is there, male child. 2 set of chromosomes, that is 2 set, diploid, female. Now understand how it is they are born. For example, in the case of male, here we have male and we have female here, female. Male is always haploid, the female is always diploid. Okay. Now they produce the gametes. Okay. We have the gametes here. We have the male gamete, and we have the female gamete. Okay. The male gamete is haploid again. This also is haploid. Now students, you tell me in the chat, if we have to go from haploid to haploid, do I write here mitosis or meiosis? Tell me. Do I write mitosis or meiosis? Tell me now. Here obviously I will write mitosis. Why? Because there is in the case of mitosis there is no reduction division. No reduction division. The ploidy will remain the same. But in the case of female, we'll have here meiosis. Meiosis. Now the two gametes will fuse together and we get the offspring. And the offspring will always and always be, as a result of fertilization, the offspring will always and always be female. Always and always be female. Why? N plus N is 2N. And 2N is always female. 2N is always the female here. And this female right here can be a worker. It can be a worker or a queen bee. Worker bee or a queen bee. Always and always and always. It is fixed for sure. Now you will ask me, sir, how are the males produced then, sir? How are the males produced? Males. Understand, this female gamete right here, it could be egg. Okay. This female gamete right here will directly undergo. This female gamete right here will directly undergo a process called as parthenogenesis. Parthenogenesis. As a result of parthenogenesis, it will lead to the development of a male. Now, male will also be N, and the male is called as what? The male is called as a drone. The male is called as a drone. Male is called as a drone here. Male is called as a drone. Now, students, I'll teach you something very interesting right now. Very interesting. Here, now tell me, students, can this drone understand? Can this drone right here, male, can he give birth to a male child? Obviously not, because this male here will produce the male gamete by mitosis eventually and will always give rise to a female only. So can I tell this male right here, after growing up, produce the gamete, always result in a female? Yes. So can I tell there is no possibility of a son? No son. No son. Okay. Now students, tell me, this male gamete right here, this male drone right here, does he have a father? Is there a father there? Tell me, students. Here it is directly developing from a female. So can I tell technically there is no father and only a mother? So can I tell no father? No father at all. Okay. Now students, tell me, this female will eventually, as a result of parthenogenesis, can it give rise to a male eventually? As a result of parthenogenesis, this female? Yes. So can this male, even though this male does not have a son, can I tell this male right here, can he have a grandson? This female will eventually give rise to a male. Parthenogenesis, right? Parthenogenesis. So can this male eventually has a grandson? Yes. Can have grandson. Can have grandson. And this male right here, even though there is no father, does this male have a grandfather? Male grandfather? Yes. So even though there is no father, there will be grandfather. Grandfather will be present here. Grandfather will be present. Amazing, no? Amazing, no? Yes or no? Yes. So there is no son, there is no father, there is a grandson and grandfather. So with that being said, solve this question, all of you. Time starts now. Tick, tick, go. All of you. Solve this question. Solve this question, all of you. Solve this question. Given below are two statements. One is labeled as assertion reason, okay? In Honeybee, the population, the sons do not have a father but have grandfather. Yes. Haploidity in drones of honeybee is due to parthenogenesis, while fertilization results into a female bee. Yes. Because of this reason only, we always have grandfather and grandson, but not father and not son. Yes. So it is true. An explanation. Yes or no? Done. Next question, students. In animal kingdom, along with XX and XY mechanism, many other variants of chromosomal sex determination mechanism have been described as such. That is XXXO. So XO mechanism was grasshopper. This was grasshopper. Okay. ZW was your birds. Yes, it was birds. ZO was butterfly or moth. This was usually butterfly or moth. Okay. Mechanism and haplodiploidy mechanism was seen in the case of where? In the case of honeybee and wasp. In the case of honeybee or wasp, honeybees or wasp or ant also. Okay. Which of the following represents respectively? So grasshopper, bird, moth, wasp. Option number 2. Done. Okay. Next question. Select the correct statement with respect to mechanism of sex determination in the case of grasshopper. It is an example of female heterogamety. No, male heterogamety. Male produces two types of gametes, either with or without X. Yes, correct. The total number of chromosomes, autosomes and sex chromosomes are same in both male and female. No, male has less. All egg bears an additional X chromosome besides the autosomes. Yes. So, what is the answer? B and D, right? B and D. So, option number 1. Yes, done. The XO type of determination is seen in the case of just now we learned in the case of grasshopper. XO is seen in the case of grasshopper. That's it, student. That's it. So that was the simple concept of sex determination. You can get many questions. Did you see that? You can get questions from 2022, question from 23, question from your 24. Every single year. Every single year. 2024. So 2024, 2023, 2022. So almost every single year, one question has been repeating year after year in the terms of sex determination, in the case of pleiotropy, in the case of your diseases, in the case of your especially the monohybrid cross or dihybrid cross, especially the incomplete dominance. So when you do the questions, that is PYQ, you get a idea about which part of chapter one question we can expect. And right now all of you are clear with the concept, all of you know the beautiful concept of sex determination right now. Okay, so let's go to the next question called as mutation. After mutation we have pedigree. After pedigree analysis we will take a small break and then we will do the entire diseases also in today's class only. Okay, now friends, next concept is mutation. Very simple. Now students, mutation usually questions do not come. Let me tell you why. Because about mutation you will learn in detail in the next chapter, not in this chapter. Next chapter, that is molecular base of inheritance, where there you learn exactly about what is a point mutation, what is frame shift mutation, right? Exact point to point you study the next chapter, not here. Okay, I'll tell you what are the main points here. Okay, what is mutation? Mutation is nothing but sudden inheritable change or a change in the chromosome, a small change in the gene, that is mutation. Okay, and students, one small, I'll tell you a hard fact. Mutation, okay, don't think that because of mutation you will get superhuman abilities. For an X-Men you will become, not possible. The maximum you will get because of mutation is cancer. Okay, so don't think that because of mutation I can get all the amazing powers in the world, not possible. Mutation will lead to cancer most of the times. Okay, unfortunately. Okay, there is a change in alteration in DNA sequence. Alteration in DNA sequence is called as your mutation. It is called as mutation. Okay, In addition, recombination. In addition to recombination, mutation is another phenomenon that leads to variation. Okay. In chapter 5, that is molecular basis of inheritance, you will learn more about it there. Okay. That is what is deletion, what is your insertion or duplication. So deletion of a chromosome, so deletion of a gene or deletion of a and addition of a gene, everything can lead to your entire mutation. Okay. See, normal set of chromosomes, translocation can happen, disease. Deletion, entire thing can be deleted, entire nucleotide sequence can be deleted, okay. Or entire base pair can be inserted here, again mutation. Or this is called as point mutation. See, in the case of point mutation, this is your point mutation. Point mutation. In the case of point mutation, do you see one of the nucleotide can be completely replaced, thymine to cytosine. Frame shift mutation, one nucleotide has been removed. Imagine if this nucleotide has been removed and this entire frame moves front, that is called as frame shift mutation. And all of this mutation, students, all of this mutation, the main cause is, one of the main causes is UV rays, right? UV rays, or there are many carcinoma or cancer causing agents or mutagens. Many mutagens are present. Okay, so be very careful of sunlight also. Okay. They have given you one example here called as phenylketonuria. See, point mutation. The classical example of point mutation is what? Sickle cell anemia. Sickle cell anemia is a classical example. It is a classical example of point mutation. Point mutation given right here. Okay. Now, students, I will teach you a few things. Right. In mutation, there are two main categories, students. In mutation, that is change in chromosomal number, there are two main categories. Understand? Write down. Okay. Change in chromosomal number. Change in chromosomal number. There are two main categories here. What are the two main categories? Understand. Change in chromosomal number. There is something called as aneuploidy and euploidy. Aneuploidy and euploidy. Aneuploidy and euploidy. In the case of euploidy, the NCERT example they have given you is polyploidy. Polyploidy. Now, what exactly happens in the case of your euploidy? In the case of euploidy, entire set of chromosome is changed. So what will happen? Your 2n can become 3n, 3n can become 4n. So entire set of chromosome is changing, and this is mostly seen in the case of plants. That is euploidy. Example is ploidy given you NCERT. But what exactly happens in the case of your in the case of aneuploidy? In the case of aneuploidy, there is a change in the single chromosome numbers. One chromosome can be deleted, one chromosome can be added. Here entire set, here a single chromosome change is happening. A single change is happening here. Single chromosome change is happening. Now there are two examples given here. It can be further divided. Into something called as hypoploidy or hyperploidy. Hypoploidy and hyperploidy. Now, what is the difference? Students? What is the difference? In the case of hypoploidy, there is a decrease in number of chromosome number. Decrease. In the case of hyper, there is increase in number of chromosome. Now, students, I'll give you a quick example. The quick example which I have to tell you, all of you know this example. In the case of decrease in number of chromosome, all of you remember Turner syndrome. Turner syndrome. XO type, XO arrangement, Turner syndrome. Decrease in number of chromosomes. Yes or no? Decrease in number of chromosome. Yes. Here we have something called as Down syndrome. Down, that is trisomy of number 21. Here we have XO type, XO. Then we also have Klinefelter syndrome. We also have Klinefelter syndrome here. That is XXY. One additional X chromosome, hyper, increase number of chromosome. So this is the chromosomal change in number. Okay. So we have euploidy and aneuploidy and euploidy. Okay, aneuploidy and euploidy. Now students, the next concept is called as pedigree analysis. Not dog biscuit, not dog biscuit. So pedigree analysis. Now students, in the case of Mendel's experiment, every single time we knew there was a change in the plant or disease was there in the plant, you can easily tell from which plant it is coming. You can see the generation on the garden only, you can tell. Why? Because the life cycle is very short in the case of plants. You can easily find the disease. But in the case of humans, where the lifespan is very large, you cannot follow the same thing. In the case of humans, it is very difficult to do the same thing. So in the case of humans, if you want to find a genetic disorder, there is a small hack, or there is a small trick, or there is a small mechanism that is called as pedigree analysis. See here. The idea that disorders can be inherited has been prevailing in the human society for very long. Okay. What is the basis? The basis is that heredity of the certain characters are transferred from the parents to the offspring. Okay. Since it is evident that control cross that can be performed in the pea plant or other organisms are not possible in the case of humans, the study of family history about the inheritance of a particular trait provides an alternative. Such analysis of traits. So what are we doing? We are analyzing the traits: tall, short, dwarf, color of the flower. In the case of humans also we have certain traits: eye color, height, arm length. There are many traits. Okay, in several generations of a family is called as pedigree analysis. In pedigree analysis, the inheritance of a particular trait is represented in a family tree. So you can see a family tree here. You can see an entire family tree, and this family tree, students, this family tree will help you to identify. This family tree will help you to identify which trait is present in which human, and is it transferring or not, or a pattern of transfer. Based on the pattern of transfer, we have autosomal disease and we have sex-linked disease. In autosomal, we have autosomal dominant, autosomal recessive. In X-linked, we have X-linked dominant, X-linked recessive. Okay, so all that is there in the case of your pedigree analysis. So with the help of pedigree analysis, one can try to find which disease is being inherited from the generation to generation. Okay. Now students, there are certain symbols here. So in examination, if you look at your previous year paper, any previous year paper, very rarely they will ask you to decode a pedigree analysis. In 90% of the cases, the question will come from two main topics. The first main topic is the entire symbols, and the second thing is that the only two symbols or the only two pedigree family tree which they can give you is these two. One is your autosomal dominant, other one is autosomal recessive. Autosomal dominant, the example is called as myotonic dystrophy. Autosomal dominant myotonic dystrophy here, right here. Autosomal dominant. The other one is something called as sickle cell anemia, that is autosomal recessive. Autosomal recessive. So students, before you try to solve pedigree, the two main things which you need to know is the symbols as well as the two different examples given your NCERT, that is your myotonic dystrophy for autosomal dominant as well as sickle cell anemia for autosomal recessive in nature. Okay, so learn these two, it will help you in the longer run. These two right here, and learn the symbols. See, male is square, female is round. Okay. Mating is single line. Mating between relatives is two lines. Consanguineous mating. Parents above and children are below. Usually affected is red color. Sex unspecified. Sorry, 5 unaffected individuals is diamond and 5. Sex unspecified is rhombus or diamond. Okay. So in examination they will 99% they will ask you either of these two. And I'll prove you also by showing you some questions. Okay. Now, what is that 1% or 2% or 5% where question can come, sir? That also I'll teach you right now. Okay. Now, for sometimes what happens is they can tell to ask you to identify. They'll ask you to identify if the given pedigree is autosomal dominant or recessive, or is it X-linked dominant, X-linked recessive. In such cases, there are small hacks or tricks which I have found. I will write on the board. All of you write with me. Make a copy of it and stick it on the board and look at it every single day. Only when you look at every single day and use the tricks to solve some questions, then only these tricks will be helpful to you. Otherwise, forget it. Okay. So, what are the tricks here, students? What are the tricks here? Understand. What are the tricks here? Okay. Very simple tricks which I have for you, all of you. Okay. Very simple tricks. Very simple tricks which will help you. Okay. First one is something called as autosomal. Understand. Autosomal. First one is called as autosomal. Autosomal. Now, under autosomal, we have something called as autosomal dominant and autosomal recessive. Okay. We have autosomal dominant, and autosomal recessive. Now, what exactly happens in the autosomal dominant, friends? In the case of autosomal dominant, they do not skip generation. They don't skip generation. Nope. Here they skip generation. Here they skip the generation. A disease can skip the generation here. It can skip generation. It can skip the generation. Now, students, in the case of autosomal dominant, affected parents can have unaffected children. Unaffected children. See here. In the case of your affected parents can have unaffected children. Affected parents can have unaffected children. It is not skipping generation. This generation, this is the same generation, right? Generation is same. It is always there. So it does not skip generation. Affected parents can have unaffected children. Now here it's opposite. Is the opposite here. That is unaffected parents can have affected children. Unaffected parents can have affected children. See here. Unaffected parents can have affected children. Unaffected parents can have affected children mostly. Okay. So here, see, unaffected parents do not have any disease. Autosomal recessive, unaffected parents can have affected children. Unaffected parents can have affected children. It is skipping generation. This generation, there is no disease. Skipping generation. Okay. That is for your autosomal. Understood? Autosomal. Now, next one is called as X-linked. In X-linked also, students, under your X-linked, also we have the autosomal dominant and autosomal recessive. sorry, sex-linked dominant and sex-linked recessive. Sex-linked dominant and sex-linked recessive. Understand now. Now here also, in the case of dominance, one second students. Okay. Now, in the case of autosomal dominant, the disease will never go from father to son. First main point. Never from father to son. It will never go. It will never grow from father to son. Okay. Understood? Now, what exactly happens other than that? Autosomal dominant, all daughters of an affected father will be affected. Why, students? Now imagine the father is affected. Obviously the father is giving the X chromosome here, affected chromosome. So it is a very high chance that the daughter will get the same chromosome. It X-linked dominant. So this is dominant here. So daughter will also be affected here. And because the same reason, the more females are affected here. Females are affected more. Females are affected more in the case of X-linked dominant. Okay, X-linked dominant. Okay. On the contrary, in the recessive, males are more affected here. And the disease will always go from mother to son. Here, in the case of X-linked recessive, the disease will always go from mother to son. And father to daughter. Mother to son and father to daughter. Okay, father to daughter. Students, so write down all the following ones, and notes will be available on Telegram channel anyway, my Telegram channel or NEET English Telegram channel. So download these notes, solve such questions, use the tricks, and then see the magic happen. Okay. So this is there is one more Y-linked also. Y-linked, I forgot. Y-link is very simple. In the case of Y-link, only males. Y-link is male to male. Father to son, always father to son. Only males are affected. Why? Because Y chromosome. Only Y chromosome. Females do not have Y chromosome. So only one possibility is there. No other possibility. Yes or no? No other possibility is there. Okay, done. That is done. Okay, so Marden dystrophy, autosomal dominant, the only example, and autosomal recessive is going to be sickle cell anemia. So I hope you're able to follow this. Any doubt you can ask me in the chat. I'm looking at the chat right now, all of you. Srinath, Sony, Sneha, Ravindrani, Techno Blade. Okay, Oxytocin, Vishnu Priya. No doubt, right? No doubt. Amazing. So that was your pedigree analysis, done and dusted. So there is a simple question. See how many can simple. After this, I'll give you a break. Which of the following symbols represents the mating between relatives in human pedigree? So mating between relatives is consanguineous mating, right? So two lines. Next question. Which of the following occurs due to presence of autosomal linked dominant trait? I've always told you, Autosomal link dominant is only one is there. Only one is there. Autosomal link dominant, there is only one example, that is myotonic dystrophy. Always myotonic dystrophy. Only one example. Next question. A father will never. Here also. Friends, here also, in the X-linked recessive also, students, X-linked recessive also, father to son will never go. See, it's X-linked always. In X-linked, it never goes from father to son. Never go from father to son. Why? Because in X-linked disease, the X chromosome in the son is always coming from mother. Right? It is always coming from mother, not from father at all. Okay, not from father at all. Father to son, never. Father to son, never. Father will never pass the disease to hemophilic to his son. Yes. Hemophilia is X-linked recessive trait. Hemophilia is a X-linked recessive trait, friends. X-linked recessive trait. Hemophilia is X-linked recessive trait. All of you smash that like button first of all. Very less likes. Tell me, what is the answer now? I am telling you, assertion is correct. Reason: hemophilia is sex-linked recessive trait. Hemophilia is sex-linked recessive trait. So students, there are two X-linked diseases given in your NCERT. One is color blindness, other one is hemophilia. Correct. Okay. So since it is X-linked recessive trait, that is why it is never going from father to son. Okay. That is why it is never going from father to son. Both are correct. Select the incorrect, match the following regarding the symbols. Each match the following 6 unspecified affected individual. Consanguineous mating, again, imposter right here. 2 lines, should be 2 lines. Should be 2 lines here, right? Study the pedigree chart given below. Okay. See, affected parents can have unaffected children. Affected parents, unaffected children. So can I call it autosomal dominant? Yes, I can call it autosomal dominant. Since it is autosomal dominant, the only example given your NCERT for autosomal dominant, what is the answer? The only example given your NCERT for autosomal dominant is myotonic dystrophy. Myotonic dystrophy. Okay. Students, now we will take a small break and then we will start the diseases, because many questions are coming up. Many questions are coming up. Can you see? Many questions are coming up for your diseases. I have given few questions here. So let's wait for a small break and then we will start with all the diseases. Okay. So there are many questions for color blindness, many questions for hemophilia. See here. Many questions for hemophilia is right here. So we will take a small break and then we will start with the color blindness and diseases. Okay. 35, 45, 55, 20 minutes break. Class resume at okay. Chalo, I'll see you all of you at 1:55. All of you eat something and come and run and come back again later on. Okay. Chalo. Bye bye all of you. Take care. I'll see you at exactly in 15 minutes. Okay. One second. Let's make it 55. Fine. I'll also eat something and come then. Come. Chalo then. Come back. We have a lot to do still. Hello students, welcome back. Welcome back after the break. I hope all of you ate something and you came. And you're ready for the part 2 now. Now, next part is hardly for 30 minutes students. Let me tell you confidently. The next part is hardly for 30 minutes. There are few questions we'll solve and all the disorders. Can we start? Can we start students? So let me just erase this first of all. Let me erase this. Now before we go to the color blindness, I'll make a complete flowchart for you. So write on the flowchart, because in examination how will they ask you questions is, they will ask you questions directly. Which is autosomal dominant? Which is autosomal recessive? Which is X-linked dominant? Right? So such questions can come in examinations. So write down a few points and the disease where they fall under, and after that you will not have any confusion. Okay. For example students, let me just take an empty slide here. Let me just take an empty slide and let me write it for you. Okay. For example, the entire genetic disorder, the entire genetic disorder can be classified into two broad categories. One is called as Mendelian disease. One is called as Mendelian disease. One is called as your Mendelian disease or disorder. The other one is called as your chromosomal disorder. The other one is called as what? Other one is called as chromosomal disorder. Chromosomal disorders. Okay, done. Now students, understand the Mendelian disorders. The entire Mendelian disorders is mainly because of a small change in the gene. That is alternation in single gene. Alternation in a single gene. Okay, done. Now here we have something called as, here we have autosomal. You have autosomal and we have one second. Okay. Here we have autosomal and we have sex linked. Here we have autosomal and sex linked. Okay, done. Now under autosomal, Again we have dominant and recessive. We have dominant and recessive. Okay, dominant and recessive. Now under the examples, now understand. Under the examples, here we have myotonic dystrophy. The only example, students, the only example of autosomal dominant is myotonic dystrophy. Myotonic dystrophy. Myotonic dystrophy. Now what about in the case of myotonic dystrophy? What about autosomal Mendelian recessive? Here we have 4 diseases. One is sickle cell anemia. Then we have phenylketonuria. Yes, we have phenylketonuria. Then we have thalassemia. We have thalassemia, both alpha and beta, and finally the one more here is, it's given your NCERT, but that's all, it's fine. 3 are enough, students. Albinism, you don't usually ask you. Albinism, they don't ask you usually, so don't worry. Autosomal diseases, genetic disorders, yes, all its autosomal disorders are genetic disorders. Now under sex linked, we have the color blindness, X mostly. We have color blindness, and apart from color blindness, there's one more disease now, students. As soon as you get a cut, you get heavy bleeding. There is non-stop bleeding, also found in the case of Queen Victoria family lineage has it. Can anyone tell me which is the disease which I'm talking about here is in sex linked, the disease which is a blood related disease where the bleeding does not stop? Hemophilia. I hope you're able to write it down. Now again, students, in the case of chromosomal disorders, also we have something called as your autosomal dominant and autosomal and sex linked. Here also we have autosomal as well as autosomal and sex linked. Now in autosomal, students, there is only a single example. There is a single example given your NCERT. Can anyone tell me? That is trisomy of 21. Anywhere you might have heard trisomy of 21, students. Many of the autoimmune disorders are genetics. Yes, Huntington disease and all. Huntington, it can easily be transmitted and everything. Okay. Now in autosomal, all of you know trisomy of 21, that is Down syndrome. Now under sex link, we have 2 one is Turner syndrome, other one is Klinefelter syndrome. That is under chromosomal disorders. So in examination, you should be able to write, right? You should be able to write exactly which will be asked where, right? For example, students, small game will play now. Now I will tell me autosomal examples you're supposed to say. Okay, so look at it for like exactly I'll give you 1 minute. Look at it, then I'll ask you a question. Tell me once you're done, we can start the game. Quickly tell me. Done? Okay. Tell me now, students, the example of sex-linked recessive diseases. Example, go in the chat, all of you. Sex-linked recessive diseases. Sex-linked recessive disease. So you can directly go here. Sex-linked recessive disease, color blindness and hemophilia. That is Mendelian. If I ask Mendelian is these 2 examples. Okay. Now I'll ask you one more example. Tell me the example of Mendelian autosomal dominant. Mendelian autosomal dominant. A single example is given. Mendelian autosomal dominant. A single example is given, your NCERT students. A single example is given. MD. Exactly. Myotonic dystrophy. Okay. Tell me the example of chromosomal autosome disease. Chromosomal disease, which is an autosomal disease. Go. Let's see how many of you are able to tell now. Chromosomal dominant autosomal disease. Let's see how many of you tell. You will tell me, sir, Down syndrome. Okay. Last question. Tell me Mendelian autosomal recessive disease. Mendelian autosomal recessive disease. Autosomal recessive. You will tell me, sir, sickle cell anemia, phenylketonuria, and thalassemia, as well as thalassemia. Albinism also can be asked. Yes. Okay. So such questions can come in examination, so be careful. Now understand color blindness. Okay, color blindness, very simple. First of all, color blindness, all of you just now learned, color blindness is under sex-linked recessive disease, right? It is sex-linked recessive genetic disorder. Done. Okay, first point is already clear to all of you. Now what exactly happens in color blindness? In the case of color blindness, inside the eyes there is something called as rods and cones. The rods and cones gets damaged, or they become defective. Because of a small mutation. In the X chromosome. Okay, so mutation in the X chromosome can lead to defect in the rods and cones. But the surprising thing here is the males are generally more affected here. The males suffer more compared to the females here. And usually females are carrier. Now because of this color blindness, I am not able to tell between two main colors. Which is red, which is green. I am driving my car and I am going. I am looking at the signal, everything looking grey to me. I cannot tell which is red and which is green. That is because of color blindness, and usually males are more affected. And it is a sex-linked recessive disorder, X-linked recessive disease, mutation in the X chromosome. Done. See here, NCERT is given here for all of you. Now, it is sex-linked recessive disorder. All of you know this one line. Very important line. Okay. Due to the defect in the red or green color, green cone of the eye, resulting in the failure to discriminate between red and green color. You cannot tell which is red, which is green. Not possible at all. Okay. This defect is due to the mutation in the certain genes present on the X chromosome. And usually males are more affected. See, 8% of the males and 0.4% of the females. Okay. This is because the genes that lead to green-red color blindness are present on X chromosome. Males have only one X, female have two. All of you know. Okay. This is very important. The son of a woman who carries the genes has a 50% chance of being colorblind. Okay. The mother is not herself colorblind because the gene is recessive. This means that effect is suppressed by a matching dominant phenotype. So basically all they're telling you is it is a recessive disorder, and the chances of female becoming colorblind is very rare. The only way the female will become colorblind is how? A daughter will not normally be colorblind unless her mother is carrier and her father is also colorblind. That is one of the only cases where the female will become colorblind, unless both the parents are colorblind. If both parents are colorblind, obviously daughter will be colorblind. Okay. Now students, in examination, how will they ask you questions? In examinations, they will ask you numericals. One on the board. Let's see how many of you are able to solve such numericals. Time starts now. Go. Time starts now. If a colorblind female, right? Female is colorblind. So X. Okay. Colorblind female marries a man whose mother also is colorblind. So mother is colorblind the son will get the X chromosome from here. Okay, so the male will be like this. Male is XCY, the female is XX. Okay, what are the chances of our progeny having color blindness? Okay, let's do a simple cross. Gametes, do the cross. Do a simple cross now. Tell me what is the answer. What are the chances are progeny having color blindness? Colorblind, 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 colorblind. 100%. Yes or no? Yes or no? See, colorblind, carrier, right? Colorblind, 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 colorblind. So can I say 100%, the entire offsprings are going to be colorblind. This is how the questions will be asked in examination. Do not expect them to ask easy questions when it comes to diseases. Okay, I'll give you one more question. See, how can a female be colorblind? Okay, the father colorblind and mother carrier, we just now learned. We just now learned this one. Yes. Okay. Next question. See here. Next question, you see, all of you. Very interesting. In humans, color blindness is X-linked recessive trait. Look at the X chromosome. Okay. A colorblind man. Okay. Man is colorblind here. So XCY. Okay. Marries a woman who is a carrier for colorblind. The female is carrier. Female is carrier. Okay, carrier. Female is carrier. Okay, female is carrier. What is the probability of the children having color blindness? What is the probability that the children also being colorblind? Okay. Gametes, all of you gametes. She is a carrier, mother is a carrier. Father is colorblind, right? Father is colorblind. Colorblind man, okay. What do we get here? X, C, Y, X, C and X. Clear, students? What are the chances of the progeny is becoming colorblind? This is 100%. This entire thing is 100%. So 25 plus 25%. So these two will be colorblind, right? Colorblind. XY. This will be carrier. This female will be carrier. Well, this is going to be normal male. So the progeny of being colorblind. The progeny of colorblind is 50%. 50% is the answer. Or students, the same question, they can change also. They can change the same question into what is the percentage of females becoming carrier. Question is very simple. Tell me, what is the percentage of females being carrier? They'll give this question. Answer is what now? Answer is the female being carrier is 25%, 50%. See, this is 100% female. 100% is female, 50% is carrier. Female if they mention. Now if they ask you what is the percentage of colorblind offspring? Colorblind, see, read the question carefully. Percentage of female being colorblind. 100% female, 50% female is carrier. 50% is carrier. Now if they ask you in question, what percentage of progeny is carrier? That time 25%. That time 25% is carrier, progeny. So always read the question carefully if they are asking you for progeny or child specific, sex specific. So be very careful while you are answering these questions. Okay, so this will only happen if you do enough of practice and you will not make a mistake. Okay, so did all of you understand? So listen, read the question carefully. If they're asking for a sex of, if they're asking specifically for a progeny or for the entire and progenies or only a female or male. Okay, so read it very carefully. So what you need to do is solve all of these questions. Solve all of these questions one by one, download the notes and solve all these questions. You will get some confidence, and any questions you're not able to solve, let me know, ask in the comment section. I will reply to it with the answer. Okay, so questions are given here. Go see more questions. Do you see that? Questions, more questions, more questions. Okay, so solve all of these questions. You will get a better grip on the concept and you will not make the same mistake again. Okay, the next disease is called as hemophilia. Hemophilia is a disease of a blood disease. It is a blood disease and it is sex-linked recessive disorder. First point, all of you know. First point, all of you will know this. All of you will know this. Okay, now what exactly happens here? See students, every single time you get a cut, every single time you get a cut. Okay, when you get a cut, after some time this blood platelets will completely fuse and blood flow will stop. But in the case of hemophilia, a small cut can lead to a heavy loss of blood. Why? Because there is one enzyme or one particular protein is missing. That is a single protein that is part of your cascade of protein, that is AHG protein. The AHG protein is missing. Okay, a simple cut will lead to non-stop bleeding. The heterozygous female carriers for hemophilia may be transmitted the disease to sons. Always. Okay, very important line. We'll come back to this line again later on. The family pedigree of Queen Victoria shows a number of hemophilic descendants and she was a carrier to the disease. Possibility of female becoming hemophilia is extremely rare again. It is extremely rare. Females becoming again is very rare. Females are usually carriers here. So understand the concept, then I will teach you again numericals. Numericals will come. See here. Okay. Entire thing, if you read the same thing, what have we just told? Okay, what I have just told you is right here. Now see this question, all of you see this question. All of you see this question. A normal girl, okay, very good question, very good question from NEET 2022. A normal girl whose mother is hemophilic. Okay, so mother is hemophilic, right? Mother is hemophilic. Mother is hemophilic. XX. This is the mother. This is the mother. So daughter will get one chromosome from here. Okay. So daughter is a carrier right now. Daughter is a carrier. So daughter is normal, but she's a carrier. Okay. Mother is hemophilic. Oh. She marries a male with no ancestral history. So male is normal. Okay. Male is normal. Male is normal. Okay. Hemophilia. What are the possible phenotypes of the offsprings? Students, tell me the gametes here. We get XH. X, we get X and Y. Simply do the cross now. Simply do the cross. What do we get? XH and X, X and Y. This is going to be a carrier. Again a carrier. One second. Normal. This is going to be normal. This male would be hemophilic in nature. A normal male. A normal male. Okay, so what are the possibilities? What are the possibilities? Okay, hemophilic son and hemophilic daughter. Do you have hemophilic daughter anywhere? No, gone. Hemophilic son and carrier daughter. See, hemophilic son and carrier daughter. Okay, normal daughter and normal son. Normal daughter, normal son. Okay, normal son and hemophilic daughter. See, you do not have a hemophilic daughter anywhere. This also is not possible. So only possibility is B and C. So option number 2. Option number 2. B and C only. So are you getting an idea how the questions can be asked in examination for this chapter? All of you tell me. You will always get a question from Pleiotropy. Always get a question from polygenetic inheritance. Sometimes linkage. Right? Diseases, if they come in this format, they'll come. Okay. Beautiful question. Okay, see this question now, students. See this question again. Mary's father has hemophilia. Again, very good question again, all of you. Students, trust me, it is very difficult to get such questions. I have to go find these questions for all of you. Okay, so if you are putting so much of efforts, equally effort should be put by you also. So become serious. You have hardly few days left for examination. If not serious now, when? Right, so become serious, solve these questions. This is the only difficult part of genetics. Otherwise, genetics is actually very easy. Okay, next question. Mary's father has hemophilia. Okay, Mary's father has hemophilia. Okay. Mary's father has hemophilia. Okay, has father has hemophilia. So this is Mary now. Mary will get this X chromosome from here, and this one from mother. Okay, but her husband also is hemophilic. Husband also is hemophilic. The reason I took it as X is because it doesn't say anywhere Mary is hemophilic. So generally, thumb of the rule, she has to be normal. Okay. Okay. Mary's father has hemophilia, but her husband is also hemophilic. What are the chances that other daughter having this disease? Okay. Tell me the answer now. Tell me the answer. Did all of you understand? Mary's father has hemophilia. This is the father. This is the father. Okay. One X chromosome is coming from there. Generally, they are not spoken about what Mary's condition is. So we are considering her as normal only. Normal only. So XNH, XHY. So here, if you notice, only one individual is affected. This one. Look at that. Hemophilia. Carrier, normal, hemophilic. Hemophilic. The question is what? The question is saying daughter. Daughter. So only one daughter is hemophilic. So answer is 25%. Now if I change the question, to what progeny, what percentage of progeny is hemophilic? Tell me the answer now. What percentage of progenies are hemophilic? Small change in the question. Tell me now answer. Tell me now answer. Simple. Again, the concept remains the same. The concept remains the same. Now tell me what percentage of progeny is hemophilic now? 25, 50% is hemophilic. 50% is hemophilic now. See, hemophilia, hemophilia. Both of them are hemophilia. X-linked disease. X-linked disease becomes 50% that time. That time it becomes 50%. 50% hemophilia. Okay. So question can change based on what they'll ask you. So read the question very carefully. Read the question very carefully. What X not 75? 50%. 50% hemophilic. Hemophilic is 50% right now. See here. This is normal. Carrier. Hemophilic, carrier, normal. Now I'll ask you one more question. What percentage of sons are normal? Simple question again. Tell me, what percentage of sons are normal? Son is normal or sons are normal? So you'll see here, this son is hemophilic, this son is normal. So 50% are normal, 50% are hemophilic. Answer is 50% that time. Answer is 50% that time. Simple. So read the question carefully, what they're asking you. Based on that, mark your answer. Don't be in hasty condition. Okay, done. The next question is, concept is going to be sickle cell anemia. Students, are you getting an idea how the questions can be asked? Yes or no? Tell me. All of you. Pooja, no, wrong answer again. Not 25 again. How can 25 again, students? Sons 100%. 50% are normal. 50% are affected. How 25? How 25? 25 is not possible. Okay, I'll ask you one more question. Tell me, what percentage of offspring here? What percentage of offspring is carrier? Is carrier now? Tell me. One more question for all of you. Simple maths, okay? Simple maths. What percentage of offspring is carrier here? Carrier. If anyone tells 50, no, now let's see. Carrier. What percentage is carrier? So next disease is called as sickle cell anemia. Very famous question. Under all your diseases, many questions have come from sickle cell anemia only. Okay. Sickle cell anemia is a deadly blood disease where the quality of blood is changed. Quality of blood is completely changed. Okay. Changed. So inherited blood disease, autosomal linked recessive disease. See, autosomal linked recessive disease. How do we know that, students? How do we know that? We wrote it here, students. We wrote it here. Autosomal recessive sickle cell anemia. Sickle cell anemia. So don't forget this table. This table is going to be very, very helpful to you. Okay. So what exactly happens in sickle cell anemia, students? There aren't enough healthy red blood cells to carry out the oxygen throughout the body. So there is a decrease in the quality. There is a decrease in the quality of red blood cells. The quality is gone here. Quality is gone. Okay. A person has abnormal hemoglobin, that is a normal biconcave hemoglobin becomes sickle shape here. It becomes sickle shape. Mutation is happening here. Mutation in the HB gene. That is the main important here. Mutation in the hemoglobin chain is happening. Now what exactly is the mutation here, students? What exactly is the mutation here? See this. If you look at the normal hemoglobin gene, it says here HB, here it says GAG to GTC, CTC. Okay, and that becomes GAG and we get glutamic acid, normal condition. In the abnormal condition, the A is replaced by adenine is replaced by thymine. Can you see here? Adenine is replaced by thymine. Because of that, here we have A. Because of CAC, in the mRNA we have GUG, and GUG codes for valine. So in the beta globin chain, this is called as beta globin chain. At the sixth position, instead of glutamic acid, we have valine. And because of this small mutation, because of this small point mutation, there is a change in the shape of the red blood cells. From the biconcave to sickle shape, the change takes place. Sickle shaped. And because of this very same reason, the person is suffering from sickle cell anemia because of a point mutation. Now students, you need to understand here is one more concept, that is a normal person will have two genes. One gene will have two alleles. That is, a person will have HBA and HBA. Both are normal. Both the alleles are normal. This is normal condition. This is in the normal condition. In the case of a carrier person, one of the allele is affected now. HBA, HBS. In the case of infected or sickle cell anemia person, both the chains are HBS and HBS. Or HBC also you can write. So S is affected one. Sickle cell anemic one. Okay. This is very important to solve the questions. Now, the question to all of you. Do you want to solve a numerical again? Do you want to solve numerical on the sickle cell anemia also? You know, we can skip it if you don't want. If yes, here's a numerical to all of you. Here is a numerical to all of you. What is the question? In a cross between a male and female, both heterozygous for sickle cell anemia. Both are what? Both are heterozygous. HBA and HBS. One. Other one is also what? HBA and HBS. What are the gametes? HBA. What is the percent of progeny will be disease, right? Do the cross again. Do the cross, all of you. HBA, HBS, HbA and HbS. Okay. Heterozygous condition. Carrier. This person is also carrier. Out of the 100%, this is normal. Carrier, and this is sickle cell anemic person. Sickle cell anemic person. So what is the answer? 25% is the answer. 25% is the answer. Now I'll ask you one more question. You can do it on your free time. The question is very simple. The male, let's say this is male and this is female. I am changing the question to male here being, the male is here completely infected. The male is completely infected. Now I'm changing the question, so this will become S. Now your job is to do what? Your work is to find the infected progeny percentage now. Not now, you can do it later on if you want. Simple question, right? So tell me the infected progeny. Tell me the infected progeny of the following parents. Okay, that is the question to all of you. Or sometimes you get simple questions. Sometimes you get simple question like this. There's a change in the gene pool of the beta globin chain. Sickle cell anemia, okay. In beta globin chain, valine is place of lysine? No. It is a point mutation, yes. In normal gene, U is replaced by A. Tell me students, is U replacing A or is A replacing T? A is replacing T. Yes, A is replacing T. So this also is not. So what is the answer? Answer is A and C. A and C only. A and C only here. See, in which disorder change of a single base pair of the beta globin chain results in the change in the glutamic acid to valine? Sickle cell anemia. NEET 2023. NEET 2023 question. Done. Now next disease is called as phenylketonuria. Very simple, very easy. What exactly happens? Listen to me very carefully. See, in normal person what happens? In the case of normal person, phenylalanine, because of the enzyme present in phenylalanine is converted to tyrosine. Okay. Now because of mutation, the enzyme is not produced. No, enzyme is not produced. Phenylalanine hydroxylase is not produced at all. If phenylalanine hydroxylase enzyme is not produced, tyrosine will not get, phenylalanine will not get converted to tyrosine. Not converted. If phenylalanine is not converted to tyrosine, what happens? The concentration of phenylalanine increases. If this is increasing, it will take an alternative pathway. Because of the alternative pathway, phenylalanine gets converted to phenyl pyruvic acid. Now this phenyl pyruvic acid gets accumulated. It gets accumulated inside the blood, right? Accumulation of these in the brain results in the mental retardedness. Also causes skin pigmentation. It causes, hair loss. It can also cause what? It is also excreted through the urine because of the poor absorption by the kidney. Also released as a urine because of the poor absorption by the kidney. So this is phenylketonuria. Done. Same thing written here. You can go through this once. Okay. Next disease is called as thalassemia. Again, students, thalassemia is also a blood disease. But unlike sickle cell anemia, which was a quality matter, here there is a quantity matter. Quantity. Now what do I mean by quantity? In the case of thalassemia, the entire chain of alpha hemoglobin, entire chain is missing. See, if you remember the chapter biomolecules, there I teach you something called as tertiary structure of protein. All of you remember? In tertiary structure of protein, we learn that hemoglobin is an example. And we also learn that hemoglobin has 4 chains mainly. 2 alpha chains and 2 beta chains. So in the case of thalassemia, the chain is only missing. The entire alpha chain is missing or entire beta chain is missing. Now what is the cause of this missing? Let me tell you now. Okay, what exactly happens here? In the case of alpha thalassemia, on the chromosome number 16, the two genes are affected, that is HB2A and HB21, HBA1 and HBA2. Because of these two genes defect, the entire alpha chain is missing. If the alpha chain is missing, it is called as alpha thalassemia. If beta thalassemia, in the case of beta thalassemia on chromosome number 11, see, very important, 16 and 11, on chromosome 11, HBB gene is affected. Because of this abnormal gene, the complete beta gene is missing. So abnormal HB will affect beta globin chain. Gone. So in alpha thalassemia, alpha chain is missing. Beta thalassemia, beta chain is missing. That's all. That is all, students. That is all. Okay. So it is autosomal linked disease. All of you, recessive disease. All of you know. Blood disorder. Unaffected carriers. Can be carrier again. Because of mutation or deletion. Reducing the rate of synthesis of beta globin chain, alpha and beta chains. Okay. Alpha and beta thalassemia. What happens? In alpha thalassemia is controlled by two closely linked genes HBA1 and HBA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of four genes. The more genes affected, the less alpha molecule produced. So more genes are affected. Alpha thalassemia, alpha chain is missing. Beta thalassemia, beta chain is missing. Done. Very simple. See here, this line is very important here. Thalassemia differs from sickle cell anemia. That is, former is a quantitative, that is thalassemia problem, and synthesis too, a few hemoglobin change. Okay. Quantitative and the problem, and the latter, which is the latter? Latter is sickle cell anemia, is a quality problem. It's a sickle cell anemia is a quality problem. See the question here. Sickle cell anemia and hemophilia are autosomal dominant, students. Is it true? Are both autosomal dominant? Yes or no? Tell me. All of us know hemophilia is X-linked. All of us know hemophilia is X-linked, right? X-linked. And sickle cell anemia is also autosomal recessive. Autosomal dominant is only one. So statement 1 is wrong. Statement 1 is wrong. Okay. Statement number 2, sickle cell anemia and hemophilia are blood disorders. Yes, it is a blood disorder. So 1 is incorrect, 2 is correct. Done. Option number 4. Option number 4. Done. So when you write the entire flow chart, becomes very easy to solve questions. We all of us know autosomal dominant is only one disease, myotonic dystrophy. All of you know that. All of you know that. Okay. Students, every single time you have a doubt in such questions, always remember this flow chart. It will help you in a very long run, students. This flow chart will help you in a very long run. So please write this and keep. With chart. Here. So write this, not this. This is the genetics, pedigree analysis. Where is the chart? Here. So write this and keep in your book. Every single time such questions arise, you can always look back to this and easily solve the questions. Easily solve the questions. So always keep this in your mind, all the entire thing. Okay. This question, see, which of the following is not a autosomal recessive disease? Not a autosomal recessive. Just now I told you, which is not a autosomal recessive, students. All of us know hemophilia is X-linked. Hemophilia is X-linked disorder. Hemophilia is X-linked, right? So it's a NEET 2024 question, students. This year NEET paper. If you knew the entire table, this question would have been very easy for you to solve. Hemophilia X-linked, colorblind X-linked recessive disease, X-linked colorblindness. Okay. So such questions are very easy now. All of these examples are sickle cell anemia, phenylketonuria, thalassemia. All three are examples of your autosomal recessive, right? Autosomal recessive disorders. Autosomal recessive disorders. Now students, we come to chromosomal disorder. Chromosomal, again, very simple to understand, very easy to understand. All of us know, I told you already. Aneuploidy, I already taught you. What is polyploidy, I already taught you. Done, both are done. Okay. Now you need to study 3 different diseases here. One is called as Down syndrome, autosomal. Turner syndrome, Klinefelter, sex-linked. They are sex-linked, both are sex-linked. Now for understand, in the case of Down syndrome, what happens? It is because of, this is a karyotype. In this karyotype, can you see, on the 21st chromosome, there is an additional 1 chromosome that is called as trisomy of 21. Trisomy of 21, both male and female, discovered by Langdon Don. Okay, so 1 extra chromosome is there, 1 extra chromosome. Now because of that 1 extra chromosome, there's a drastic, drastic symptoms. For example, first one is a round head. You see here, the first one is what? See, it was discovered by Langdon Don, that's for the name. Okay, the first one is short stature with small round head. Second one is furrowed tongue is outside, partially opened mouth. Okay, palms with broad characteristic palm creases. The palm creases means palm lines, many lines on the palm. And fifth one, that is physical, psychomotor and mental development is retarded. All the following 5 points you need to remember. Questions will come directly from here. Nothing is difficult here. Nothing is difficult. See here, broad face, flat back on the head, many loops on the fingertips. Many loops will be here. Okay, palm creases, big and wrinkled tongue, congenital heart disease. They will always have a heart disease. Friends, question come from even this thing also, remember. Question come from even the diagram also, learn the diagram also. Okay, congenital heart disease also can happen. Next one is something called as Klinefelter syndrome. In the case of Klinefelter syndrome, there is one additional X chromosome, extra gift. Okay, that is usually the males are XY. In the case of Klinefelter syndrome, it is XXY. So usually we have 46 chromosomes, here 47 chromosomes, 47. Down syndrome also 47 chromosome. Okay, hyperploidy. See, what are symptoms here? Symptoms is again very simple. Such individual has overall masculine development, however feminine development, it is development of breast gynecomastia, is also expressed in the males. It only happens in males. Such individual is also sterile, very important line. They are completely sterile. See here, diagram I'll show you. See here, overall masculine development with feminine features, sterile male. Okay, next one is called as Turner syndrome. In the case of Turner syndrome, 1 X chromosome is missing. 1 X chromosome is completely missing. Usually the females are XX. Here in the case of Turner syndrome, it is XO. So can I tell the number of chromosomes in such individuals is around 45 chromosomes now. 1 chromosome is missing, so 45 chromosomes instead of normal 46. So 44 autosomes plus 1 X chromosome, 45 chromosomes in total. What are symptoms again here? See, such females are sterile, ovaries are rudimentary, besides the other features including lack of secondary sexual characteristics. The female will be very short and everything. This is called as Turner syndrome. Okay, so here is a question. Let's see. The question says Down syndrome, all of you know, trisomy of 21. A is going to be 3. Okay, alpha thalassemia is 16, beta thalassemia is 11, and Klinefelter syndrome is additional X. So A is 3, right? A is 3, option number 2. Next question. The manifestation of phenylketonuria are given below. Choose the incorrect answer from the given options. Mental retardedness, yes. Skin rashes, yes. Excessive accumulation of phenyl pyruvate in the blood, yes. Furrowed tongue, yes. Furrowed tongue is for Down syndrome. So this is not an answer. NEET 2024. So answer is option number 4. Next question. Which of the following statements are correct about Klinefelter syndrome? Okay. This was discovered by Langdon Don? No. Such individuals has overall masculine development, however the feminine features development is also expressed? Yes sir. The affected individuals with short stature, no. Physical psychomotor, no. This is for Turner syndrome. Okay. Such individuals are sterile, yes. So B and E is right here. B and E is right here. Next question. Broad palm with single palm creases is visible a person from. Students, Down syndrome, NEET 2023 question. 23 question. This was NEET 2023 question again. So are you getting an idea how the questions can come? How the questions can be asked to you if you can solve such questions? Easy 4 marks for all of you. Easy 4 marks. Next question you see. If a female individual is small round head, furrowed tongue, already you know it is Down syndrome. Already you know Down syndrome, NEET 2022. So Down syndrome is trisomy of 21, yes or no? Down syndrome is trisomy of 21. Easy question, easy answer. So genetics is very easy. Genetics is very easy if you're learning it the right way, solving numericals. This is the easiest chapter to score the highest of marks. It is up to you to decide right now. Okay, so any doubt anywhere, always ask me. I will always clear doubts. It is very important to become more confident when solving such questions. So confidence is the key. So with that being said, students, this is your homework question. This is your homework question. Students, I will check in the comments. In the comments, I will check. Okay. That is Madurai, Pooja, Sukanya, Sneha, right? Malaya, Newton, Jasmine. I will check all of your homework question if you're writing the homework question. If there is no homework question, no more questions for you, students. No more questions for you. So I'm doing my duty here. I'm doing my job here, that teaching you the entire chapter on one go. Right? Obviously it's part 2, but I did not skip any line of NCERT. Right? So we have done the line by line of NCERT. All of you are getting a super clear information. All of you are becoming confident in the subject. Hopefully you can continue the same until the examination. Learn line by line. No one can take away any marks from you. Okay. So with that being said, a small surprise coming for all of you. There's a small surprise coming for all of you. Let's see how many of you can guess what is the surprise, because even I don't know what is the surprise. Okay, even I don't know what is the surprise. So let's see what is the surprise for all of you. Soon you'll be getting the surprise. And with that being said, thank you so much, students, for today's class. I enjoyed today's class. It was really fun teaching. It was really fun teaching in today's class. And I will see you in the next class with the same energy, with the same josh. And keep liking the video, keep sharing the video. It helps the channel grow. It helps a teacher like me to become more, recognized. Because when we are working really hard, every student needs such a teacher who will teach them every line of NCERT. So thank you so much, students, for today's class, and I will see you in the next class. Take care. Bye bye. Keep solving questions, keep studying, keep revising, and take care, all of you.